Alright, welcome to this lecture on cell biology. Okay, so first off here, we're going to start off by talking about iPS cells. Now, what are iPS cells? So, simply what this is in history there was a big, the hypothesis that when stem cells differentiated, they were stuck as differentiated cells. Well, recently people have figured out that stem cells can differentiate, but then also become stem cells once again. And these are what iPS cells are. So as you can probably guess, iPS cells then have many useful applications. So these cells can be reprogrammed by using transcription factors including OCT4, SOX2, KIF4, and c-MYC. And these transcription factors are able to move differentiated cells back into stem cells. So how we start off with this is we start off with some adult fibroblast cells. They're put into a petri dish, and we add these transcription factors to them. And then these cells are reprogrammed, and they are then iPS cells. And what can we do with iPS cells? Well, they're stem cells, so they can be differentiated yet again. But in this case, they're not going to be fibroblast cells. They're going to be whatever we decide to make them. So let's say a patient has some sort of Alzheimer's and they're losing neurons. Well, we can use these fibroblast cells, add these transcription factors, bring them to iPS cells, and then create these neurons once again for this individual. Now, what happens if the neurons are being lost because of some genetic traits? Well, then these genetic traits are going to cause neurons to keep degrading. So what we'd have to do then is we'd have to figure out a way to change these genetic traits through different genetic methods, and then we would then be able to, once the genes are fixed, we could then actually go through this process again and just add the neurons in, and then the patient should be theoretically good to go. Now, what you can also do with these is you can just get to these iPS cells, and we can just better study them. Because obviously, stem cells are very hard to come by, and what we can do with this high amount of stem cells is we can create neuron cells or any other type of cell, and in the case of neuron cells, then we're in a very good position because neurons are very hard to get to for humans and hard to study. And in this case, there are no ethical concerns. We can just create many neurons and study them very easily. Now, the issue with integrating these iPS cells into humans is that one of these transcription factors that are added is c-MYC, which is a well-known cancer transcription factor. And the issue can become if these transcription factors are continually expressed in the human. What may occur is that, in this case, these stem cells keep reproducing indefinitely, and cancer can occur in the patient. So that is a big issue. However, if we can find a way to make this work, these cells would be patient-specific, and so because they would be coming from the patient themselves, so there is no risk of the patient rejecting these cells when we integrate them through clinical trials and possibly in the future actual methods of helping individuals. So now moving on here, and we are looking at imaging in cell biology. That is the main thing that we're going to be looking at in this lecture. So looking here, this is the first kind of recorded microscope that was able to do a good job of magnification. And what this microscope was able to do, it was very simple in method, and it simply was able to magnify to about 200 times. So that obviously opened the doors for many things. Individuals could see cells and things like that now. And it opened the pathway to figuring out better imaging technology for cell biology. So now here we're looking at modern compound microscopes. So this is considered the light microscope. So this here is the light microscope. We have an objective lens up here and a condenser lens down here. The condenser lens is capable of 100 times amplification, and the objective lens is then capable of 10 times amplification. That makes this entire apparatus 1,000 times amplification. So now, an issue here is there is very good amplification of the specimen that we have down here, but resolution can be an issue, because all we're doing here is amplifying the specimen. And resolution is considered how close two objects can come together, where we see them as distinct and not connected. So in order to increase this resolution with this apparatus, we can have a lower wavelength, and we can also have a higher numerical aperture, which is broken up into a refractive index of medium between the specimen and the objective lens. So this is simply the rotating things here. You can adjust which one you want up here, and a higher value for this will allow resolution to be lower, which is better. So also keep note, the light microscope sends light from the bottom here underneath the specimen. And so here's the light source down here, and that shines through, and that's towards the objective lens and to the light detector. The light microscope operates between 300 and 700 nanometers. So now let's consider this apparatus here is very good at amplifying, but let's consider a way to get higher resolution. Now I said wavelength and numerical aperture can have something to do with this resolution, but there are also different methods of messing with this level of resolution. And one of these is, as I said, wavelength. So let's look further into detail about that. So as you may know, a wave with higher amplitudes is going to give light that is more light, I can say, and one with lower amplitude will give a dimmer light. Certain parts of cells retract light more than others, and we can use this to further amplify the way the cell is so that we can see it better. And also consider if these light waves run into each other through destructive interference, you will not be able to see anything. So looking here at the phase contrast microscope, here we're simply using constructive interference and destructive interference to get a better image. it's actually going to help with the resolution. So with constructive interference, we are going to get a lighter image, and with destructive interference, we are going to get a darker image. As I said before, if these are perfectly destructive, in this case, there would be no light. Now, what can occur is if we use destructive interference, we can end up seeing images like this. So as you can see, it's darker, and this is because we have messed with the wavelengths in a deconstructive manner and have been able to get a darker image here. And in the other case, if we use constructive wavelengths and they come together, they would create a brighter image such as this. And it is very difficult to see resolution in this case. The resolution is not optimized. Moving on here to differential interference contrast microscopy. Now what this does in a simple manner of explanation is it takes on light rays and it uses a polarizer to cut out a certain orientation of wavelength. So wavelengths can be vertical, horizontal, and in a simple scenario, let's just say we have wavelengths coming in that have a vertical aspect and a horizontal aspect. What this polarizer can do is it can take out the vertical aspect, and all you have is a horizontal aspect coming through. And this allows for better contrast. It provides a better view of the cell, and it gives a better view of the periphery of the cell. So an example here would be this here. This is an image from it, and it shows a kind of topographical view, and it's very easy to see, and it works well. So this is also considered wide field microscopy, and this is used if you want to see particular organelle or protein. So in this case, we use fluorochromes, and they allow us to look at details of the cell with better resolution. We can also see cell movements and isolate between things we want to see. So what we simply do is we use fluorochromes, which are put into certain organelles or proteins through dye, and these dyes can be special for certain things. They can attach to certain things, which allow them to attach to certain proteins or organelles. And we're very good at manipulating these dyes and making sure that they go to the protein or organelle that we want them to go to. So these fluorochromes absorb energy, and that puts electrons in the fluorochromes into a higher orbital, which is more unstable. And considering it's in a more unstable state, the instability causes the electron to drop to a normal orbital, and this releases energy as visible light, which is considered fluorescence. Taking a look here at the microscope, it uses very, very powerful light here, and it uses some ultraviolet light as well as light that we can visibly see, and it sends this light through an excitation filter, and this filters out many of the wavelengths. And what we can do is we can specifically choose a wavelength that we want to get through. In this case, it would be the blue wavelength. And what this allows us to do, from just allowing one to get through, it allows the image to be more clear with a higher resolution. So the excitation filter blocks out many of these, and we can specifically choose which wavelength we want to go through, as I said before. Now this will hit a dichromic mirror, get reflected to the object, and the object will then reflect it back up towards this detector up here, and it will go straight through the dichromic mirror. And it's allowed to go straight through this dichromic mirror because the wavelength is now longer than this one here that came in. This simply just means it's weaker, and this dichromic mirror is capable of allowing one through and one not to pass through, just considering how strong this wavelength is in frequency. Now also consider the case where we don't have a fluorochrome dye for the specific organelle or the specific protein that we are interested in looking at. Well, then we have to move on to something else. And we will run into a bit of trouble. This method here that I'm about to talk about is actually very expensive, and it can be time-consuming and difficult to do. But we must take a look at it because it works very well, and here it is. And that's a picture of fluorescent microscopy. So this is it, monoclonal antibodies. So what we do here is we inject an animal, usually a mouse. We inject it with an antigen, and this mouse will, it may not survive with an antigen injected into it, so it will try to fight it off like any other animal would. And it will fight it off with antibodies. So this mouse begins to create antibodies for this antigen, and if we continue to apply this the syringe to the mouse and inject antigens, more and more antibodies will be created. And we do know that antibodies end up coming together at the spleen area, usually. And so we can isolate the spleen, kill the animal, in most cases, which is unfortunate, but so they take out the spleen and they isolate these cells. And they know that these cells have antibodies that are of interest because they are capable of killing off this antigen. So we then grow these cells in dish, but now recognize that these would be primary cells, as I've said in the previous lecture. So they will not survive for very long. So to get away from this issue, we fuse them to immortal cells, so transformed cells, also known as cancer cells. And these fused cells are considered hybridomas, and that's showed here. So yeah, we have these tumor cells, and we fuse them with these plasma cells and create these hybridomas. And to make sure that that we isolate these hybridomas, because we don't want any extra cells here or any tumor cells lying around, we just strictly want these hybridomas. To make sure we isolate for them, we put all these cells in a medium that kills mutant mouse cells and only allows hybridomas to survive. So it ends up this thing that we put these cells on in the plate is able to kill off these tumor cells, and these plasma cells survive, and also the hybridoma cells survive. Now, when these cells begin to proliferate, realize that plasma cells will not proliferate that much because they are primary cells and they will soon die. And we will realize that some of these cells are hybridoma cells. They will blasticize, and we can then take these cells, remove them, and put them in a different dish, and grow them indefinitely. And from here you have a monoclonal antibody from these hybridoma cells, and that's what they'll produce. So it's a really good tool because these antibodies are now specific to this antigen, and we can use them for this fluorescence microscopy. So now when we use these antibodies, we first use a primary antibody to attach the protein of interest, and then we use secondary antibodies to bind to the primary antibody, and they have the fluorochromes attached. So I don't have a specific picture for this, but what happens is the antibody here is capable of attaching to a protein of interest, and then we have another antibody that is attached to a fluorochrome, and so it attaches to this antibody, and then we can then see the protein of interest. So you might be wondering, why don't we just use one antibody to attach, and we attach the fluorochrome to this primary antibody? Well, it's really expensive, and we might want to use this antibody for something else because it attaches perfectly. And obviously this entire process is very costly because look at how many steps you have to go through, and look how long this would take because you're using one specific organism. So now there's also another method called dual label fluorescent microscopy, and we can use these antibodies for many proteins and overlap pictures to see where proteins are relative to each other. And this can only be done with dead cells. So we're basically going through this process twice, or multiple times, and we simply allow these primary antibodies to bind, allow the fluorochromes to bind onto there with another antibody, and we do that for one set of protein and then another set of protein, and then we can Photoshop the proteins, well, what we see in the microscopic images on top of each other and see where these proteins are respective to each other. So now we're going to take a look at confocal microscopes. So the light beam here is very, very intense. We use a laser here, and it's directed through a pinhole to focus the light onto a certain part of the cell, considered confocality. So it goes through this process, and it hits the cell at one specific point. Now you may know that cells are three-dimensional, and there are certain planes that we can look at the cell from. Now, the best way to get good images of a cell is to differentiate the planes and look at each plane individually, and we can see which plane gives the best representation of the cell. So the simple benefit to the confocal microscope is that you can look at cells when they're alive. And what we use to look at cells when they're alive is a different sort of fluorescence. So in the case before where we used fluorochromes that we could simply attach to proteins, well, in this case we use fluorescent protein, GFP, and these are extracted from jellyfish. Scientists recognize that jellyfish could be could glow in the dark when blue light rays were shone on it, and so they isolated this GFP protein, which enabled this this thing to happen. And what they did was they linked it to a fusion protein, which is the protein of interest in whichever cell we're trying to look at. And now we fuse this this GFP this GFP and the fusion protein into a cell, and the cell will begin producing this protein with fluorescence. And now we can look at this protein get produced in the cell from this confocal microscope. And now just to take a look at an image from a confocal microscope. So this is simply a microscopic image when the planes are not separated. And this is a confocal microscope image where the planes are separated, and we look at the best plane to see the best image. Now moving on here to deconvolution microscope. So this microscope simply takes an image from all of the possible planes of the specimen, and it uses computer software to put together the best possible image. And there's an image there of what it looks like. So on this side, this is the regular image if there is no process of looking at the specific planes and using the computer processes to figure out what would look the best. And in this case, they have implemented that, and it looks much better. Moving on here to FRET, which is Fluorescence Resonance Energy Transfer. And this is simply looking at two proteins here, and we just want to see if they end up interacting with each other. So we'll say this is protein 1 and this is protein 2. Protein 1 will take in a certain wavelength and emit a certain wavelength, and protein 2 will take in a certain wavelength and emit a certain wavelength. Now, we can tell, we can see this protein 1 if we send in this specific wavelength here at 405 nanometers, and we can notice the wavelength coming out of it and catch this from imaging at 422 nanometers. Now, what we can also do is take another look at it, and if these proteins do interact, we can shine the same 405 nanometer light ray at the two of them when they're interacting, and we will see a different light ray come about, and that's 320 nanometers, simply because they are attached and this protein is now emitting. So this is the simple imaging process for FRET, and images that we can see from this include things such as this. Now moving on here to last but not least, electron microscopy. So we have here, TEM microscopy and SEM microscopy. So they both use an electron gun here as shown, and there's an anode here as well. The electrons are shot through a condenser lens and then an objective aperture lens, and it's shone through, and then the image can be better seen here at the bottom, and this is the detector here. And in this case over here with SEM electron microscopy, the specimen is here. Light rays are shone, or the electrons are shot towards the specimen and detected over here. And as shown, they bounce off in this case, and they go straight through in this case. And this ends up giving two different kinds of pictures. So in this case, with TEM microscopy, you end up with pictures like this, which are really good with like optimized resolution, and the images are high in detail. However, we do have to kill the cells to see them like this. And in this case, the specimen is stained with electron-dense heavy metals, and electrons either pass through or get rejected, or reflected, and if reflected, we get contrast from metals. So the kind of image we end up getting here is some sort of three-dimensional image, which is very interesting looking. Now the reason the resolution is so good is because the wavelength is very small, and we are operating in a vacuum, which makes the numerical aperture amount larger. Now if we want to see a specific protein with SEM, we can take primary antibody to a protein of interest, and we can link it to a gold particle that does not allow electrons to pass through. So this antibody comes in and it's attached to a gold particle, and this antibody then attaches to the protein of interest. Now, when we take images, the image, or the protein, appears to be very dark because gold does not allow anything to pass through it. And this is simply because what does not pass through is dark in the microscope, and what does pass through is light in the microscope.